Good afternoon. Let me thank organizers, Martin, for this opportunity to give this talk. I hope I can manage. I'm still suffering of the jet lag quite a bit, but anyway, let's go. Yeah, outline simply as follows. I have something about cis-regulatory modules, alignment-free approach for finding them, high-throughput SELEX data we have been using in my group, and then finding transcription, factor binding motifs, monomeric and dimeric motifs simultaneously from this large and rich SELEX data we have. At the end, I will comment briefly on long-distance dependencies within these motifs and how to present and have some final comment on simple representation of these motifs as a finite state machine. So we are here in an Institute of Theoretical Computer Science, so finite state machine is then an appropriate thing to consider. Yeah, so here is a schematic picture of what is gene regulation. No need to comment on that in this audience too much. A regulatory module is a collection of transcription factor binding sites on DNA. So no precise definition is available as far as I know. We had the discussion yesterday night. Maybe there was some conclusion achieved then, but I'm just a simple computer scientist, and I don't have any good definition. Just some characterizations or features of how these look like. So they should consist of several good binding sites of transcription factors. These modules, these sites are spatially clustered together, so there should be some denser area where there are lots of these motifs, and this is kind of a candidate for such a regulatory module. And then they are supposed to be evolutionary conserved, which would lead to alignment-based approach for predicting such regulatory modules. But I'm not speaking today on this alignment-based approach, but just direct learning from sequences without comparison between species. Here I have a simple formalization of this module structure, which will not have too much role in my talk later on, but anyway, this has been kind of underlying way of thinking what we have been doing. So module has binding sites of factors like this F1, F2, F3, F4, and each factor motif has a location within DNA, and each of these motifs have orientation so that they are in normal direction or reversed direction. So the normal ones are here on above of the DNA, and then this is reversed here. And now the problem is given a DNA sequence, predict regulatory modules on it, and then using this kind of chain model, we could find under some scoring scheme the best among the potential chains induced by the available motifs. So I come back to this in a minute. So as I said, alignment-based approach is comparative genomics, and we did some work on that some years back, and the resulting tool is called EEL, Enhancer Element Allocator, which aligned corresponding areas of genomes from different species and tried to find consort sequential patterns of motif, individual motifs there. In alignment-free case, we try to find overrepresented transcription factor binding site pairs in this talk, dimers from SELEX data. And we have just finished a tool which we call Moder, Scandinavian word, because the data comes from Jussi Taipalis laboratory in close by Stockholm. So Moder is our motif learning tool. And then the final goal of this is combine these maybe hundreds of dimer models in the scanner that finds these good chains or predicts good chains of motifs along DNA. But I'm not having any results of this yet. So the question is to model the local steps of regulatory modules, how to score the regulatory modules. We have scores for factor to DNA interaction and factor to factor interaction, and the total score of a chain of individual motifs is just a sum of if you like, affinities of factors to bind to DNA plus affinities of successive factors to bind to each other. So to use this model, we need both these factor to DNA scores and factor to factor scores.
But this score, the way it is written, the more things you add, the bigger the score is.
Yeah. So this leads to dynamic programming, and to make this converge, you need this Karlin-Altschul condition, so that it is Smith-Waterman type of dynamic programming at the end. Otherwise, it is going to happen just what you said, that it gets longer and longer. So you need both bonuses and penalties in these scoring systems to make this work. So what is happening if we eventually will be able to use this is that from these potential binding sites for individual factors, it picks up the best chain under this scoring scheme. Then SELEX. So what I'm going to talk about mostly is just these individual motif or these individual steps here, but not about this total thing. So SELEX is already mentioned here several times. Our collaborator in this SELEX is, as I mentioned, Jussi Taipale's laboratory in Stockholm, and they have been developing SELEX protocol where currently I think they take 40 base pairs long random DNA and select binding sequences from pools of this kind of random samples of uniform samples of DNA, and in several cycles they get for the given factor that has been used in this fishing, they get reads that should have occurrences of the binding motifs for that particular factor in enriched amounts. Okay, and this is this has been over years. This has been improving, so I'm not sure if they already do longer than 40 base pairs, but at least we have been using this type of data, and these SELEX generations typically can produce maybe 10,000 to 1 million reads. So it is really rich data, and quite different from the situation for de novo motif learning as compared to some 20 years back. So this is my understanding what is happening if we use let's say a factor with this type of affinity, and then this core sequence TAATCC, when it is counted from the observed reads, it has the highest count. And when you go to one Hamming step away from this core sequence, the counts are still high, but not quite as high as this here. So Teresa yesterday killed this picture totally. But I have to trust on this. All my work is based on this. So. Yeah. So if this is not true, then I don't know what sense is in all this SELEX business at all. But there could be, of course, some inaccuracies there. And of course there are lots of artifacts in these SELEX reads that you have to remove first.
Derived from one cycle or from the aggregation of all rates?
This is derived from my head. So this is not based on this is not based on any real data. This is just illustration. What I expect to happen. Okay, so here is some information what is happening. In Taipale lab they really have a big factory. So they have been producing these SELEX data sets for hundreds of factors for human and mouse. And I don't know what the total picture is a bit uncertain for me. But what is also important is that they also produce not only for individual factors, but pairs of factors, which is essential if you want to really optimize this chain of motifs along the DNA. You have to get some information how likely it is that after this factor I have this here and at what distance. And using these pairs of factors, in SELEX they can really get this information. How long? What is the spacing?
So the binding is how do you enrich? You have something that captures both proteins.
In this case, in pairs of factors. Yeah, I assume that he starts with factor 1, first generation, then next generation with factor 2, the third generation again with factor 1.
Yeah. I thought the word will suggest they make some fusion protein.
Yeah. Okay, but my guess is that this pair of factors thing is not totally symmetric. So there is a first factor and second factor that might have some effect. Yeah. So learning binding motifs from SELEX data. As we have rich data, we could consider many types of models. What and that the Moder is doing is just classical position weight matrices, but also Markov model is now possible. And not only this linear spanning tree type of Markov model, but the model Eric was speaking yesterday, where we have a real spanning tree, not only linear. Then when you look at the typical logos, quite often it is the case that this is maybe not very good example, but anyway, the number of high affinity strings is not that big, according to the logo. So here I have sketched the situation, or this is now based on real data. So this string, for example, occurs 44,000 times in the data. And here is the list in decreasing order of frequency. Why don't we just take these strings as the model? Here is the representation of these strings as a finite state machine, where I have weighted the transitions by the count how many times this data goes along each path here. So this doesn't generalize anything, but at the end, if time allows, I have a few more pictures of how this could work. Yeah. So the basic scheme what we are doing is that we want to we give seeds for the monomer models in the data, and try to learn all possible or preferred dimeric structures built from these monomers. So here is just how this dimer can happen. So it is so that the first head of this is against the tail of this, so it is HT type, or the head of this is towards the head of this, so it is HH type, and so we get these four different arrangements, and the distance D is also essential. And we model distance base per granularity, so it is really detailed. So here again, this parametrization of dimeric structures. Here are two successive motifs, and then the head of this is towards the tail of this. So it is HT type, and the distance between this is D, denoted by D. And if they overlap, then the distance is negative. So then what we especially want to model here is the deviation from the pure modular model, where the dimeric models also in the overlapping case are just products of the monomers. But Jussi Taipalis group has just recently published a paper where they studied, of course, these same problems, and their observation is that in large number of dimeric cases, the specificity of dimeric motifs differs notably from what could be expected from the monomeric motifs. So what we are doing in Moder is to represent explicitly the deviation from this pure reduction to monomeric motifs in the overlapping case of dimer. So what we do is just a mixture model where a probability of a SELEX read X, which is of length 40, for example, and contains maybe many motif occurrences. Motifs are typically maybe 8 to 13, 15 monomeric motifs. This is long, so we just represent the SELEX read in these three separate cases. The binding site of a monomer occurs in the read. Binding site of a dimer occurs in a read, and for dimer we need to know the relative orientation and the spacing between the two monomers in the dimer. Or X is just from background. So this leads to this kind of formulation, which I don't think to go through in detail. Maybe this is here is my correction term. So this is the for correcting overlapping dimer as compared to the product of the monomer components. And the log likelihood is like this, and then to solve this we just follow the path of meme, more or less. So we use EM algorithm, which now learns simultaneously for each monomer the PVM and also the spacings and orientations for the preferred dimeric pairs built from these monomers, and we have of course this mixture model has mixing parameters, and these mixing parameters reveal how popular each alternative is. This is EM converges, but to be successful you need good starting point. So we use or the algorithm needs seeds for the monomer models, but not for dimers. So it builds from these monomers to dimer structures. We are working or have been working a long time already to automate also this seeding part, but at the moment we just have to give the seeds by hand. Okay, and then here is an illustration of the resulting model for this factor. So here is the only monomer model for this Fli1, and here are the mixing parameters for all possible dimers that we have a certain range of these parameters that can be used. So for example, here we have mixing parameter value of 0.04 for dimer that is head-to-head type and has spacing 1. And this particular dimer is illustrated here. So it is head-to-head, and here there is a between one column that is taken from the background. And here is another example of it comes from here, where the mixing parameter is 0.04 again, but this is overlap of length 6 and head-to-head type, so it is this one here, and here is the correction matrix shown, so that this which is given by the data differs this much from the plane product of this in this position here. Yeah. So as you can see, there is clear differences. Most of these dimeric cases do not get too much weight in these mixing values, but there are in this case we have actually 3 quite strong cases. 2 of them have been shown here. Then I have some tests for the different a few factors we worked with. So this is this same FLE1, and this is just to illustrate that using this combined model where we have monomer and in this case 3 dimers, this together gives better R squared value when we count 10 mers as scored by the model and as observed in the SELEX data. So these are individual R squared here, and when we combine using these mixture parameters lambda, we get the highest R squared for this. And here is another one, and maybe here is a third one where we get really strong R squared at the end. This factor only has 4 different monomers, and when we combine them, the R squared is 0.99. Then so these were just kind of sanity checks using the same SELEX data that was used for learning. But here is an independent test for ChIP-seq discrimination, and this HOX B13 gets pretty good ROC values for the individual components. They are a bit smaller than for the combined model. And here is this HNF4, which gets really high ROC curve. ROC value for already for the individual monomers, but especially for their combination. So this shows that this mixing pays off. Maybe not very much in these cases, but anyway. Yeah. So to close. 3. Okay. Yeah, a few words about how to describe the affinity landscape hidden in this large SELEX data. So for this, our friend HNF4, I don't know how we got these seeds, but anyway, we used 4 seeds. Maybe it was Jussi Taipale who told that these seeds are good for this factor. But anyway, using the motor, we can find this collection of 4 monomeric models. And then no dimers in this case. Then this same thing can be described using Markov model, and then now this combines all these together so that now you can see some dependencies which are not clear from here. So that, for example, if you have JT, then it is C that should follow, etc. So as these are independent, these columns here, the certain dependency information is of course lost. So, but if I take this most frequent 13-mer from the data, it occurs 44,000 times, and go take its Hamming environment of radius 6, and then pick 23 strings according to the frequency, and represent them as a finite state machine. I get this kind of description for the affinity landscape. Now you can see that there are a kind of 4 highways here that they don't directly correspond to these 4 cases, but anyway there is some similarity which I don't yet understand fully, if this DFA really could be useful here in representing these things. But what is important here that all the dependencies that are within the highly scoring or high frequency strings, this thing, they are preserved here. There is no learning, no generalization. It is just as the data says. So what I'm aiming at here is that a motif is just a set of motif instances. And here's a sketch of algorithm how this would work. Decide motif lengths, find seeds, some locally maximal k-mers in the frequency landscape. Take most frequent k-mers in R Hamming neighborhood, or maybe you should allow some shifts because the mountains in this landscape are such as they are. Jussi Taipale calls this neighborhood Huddinge neighborhood because his laboratory is in Huddinge suburb next to Stockholm. And represent what you get as a minimal weighted deterministic automaton. So this is for 31 such strings listed here. So the count here is about 44,000 for the highest counting, and I got down to 2,200.
It's obvious that you can always turn these counts into an automaton. I mean, I can imagine that you could have that once you build the automata, it implies certain sequences must be able to occur, whereas they in fact never occur.
Say again.
It's not obvious to me that whatever list of frequencies you get, you can always build an automaton, because there's a sort of local, the automaton is local, right? Whereas the frequency count could be.
Okay. So there are two things combined here. So the automaton is just a representation for these strings, without any role of these counts. And then later on, I just each of these strings has unique path here, and I just cumulate how many times they are gone through if by the count. So it is that way.
What round of selection are these counts for, by the way? How many rounds of SELEX?
I think these come from third generation. Yeah, but I'm not sure. But it was for this factor they had 600,000 reads or something.
So that means that like 20-fold ratio in count really reflects like a 3-fold ratio in affinity, I guess, right? Like the cubic root.
You mean?
Right, between the 44,000 at the top and the 22,000 at the bottom.
Yeah.
So that's a 3-fold difference in affinity.
Yeah. This is maybe something like 5% of this count. Yeah.
But then you take the cubic root to go back to the affinity.
Okay. Okay.
Well, maybe that's why in the middle it's so conserved, because hitting even one of those reduces the affinity by more than 3-fold. Right, and that's why.
Yeah. No. Okay, but I was not satisfied with this only. So here are more of these with different thresholds of cutting these things, and you see that there is some invariance here, but can we understand what is the landscape? I'm from this. I don't know. So I should. Yep.
Question: are you constructing these DFAs so that the language that they define is exactly the set?
Yes.
Or the minimal that captures those and may capture additional ones?
Oh no. This is just really simple-minded thing. So this is no machine learning. This is just a representation for these finite languages.
So you can't generate any other sequences?
No. But this is of course the good question: how to little bit make this softer so that they work with the larger sets. And what would be the rule to do that? But anyway, we did some discrimination exercise with this, and you see that this really, as long as it can score the data in ChIP-seq, it really gives a very good ROC curve. But this needed to go to much larger automata than what I have here. So the n gives how many strings were represented there. So here I get 0.9 something as a ROC curve, but it was already 24,000 strings represented. So it is a large automaton and then totally unintuitive to see.
Did you show a higher AUC before for that factor? Didn't you show a greater AUC for that factor in a few slides before with the PWM model, or the mixing of the two?
90-something. So this is just using automaton like this. This gives the scores for each string. I was using automata like this for discrimination. Yeah. But this many strings. So I have to stop. So the goal is to make a scanner that combines factor binding models, or a scanner that takes all factors together is likely to produce too many false positives. So maybe we could reduce the set of factors to be included in a scanner by using some cell-type specific subsets. And nucleosome should be included here. Automatic seeding is missing still. And then this last thing, if we could maybe, instead of BVMs that lose lots of information, use long dependency models. Thank you.
Time for one or two quick questions.
Yeah.
Do you use the published high-throughput SELEX data?
Yes.
Which high-throughput SELEX data are you using? The one from the 2013 data?
We have something which they have published, but we have some unpublished also. Yeah, it is mixture of which I am not totally aware of. Yeah. So our collaboration with Taipale is that we meet maybe 8 times a year and try to do over email many things, and it is not always so easy to get the data.
So is there an issue to run EM on this number of millions of sequences? I mean, I think Teresa mentioned yesterday that...
We have used very, very good seeds. So it converges quite rapidly, and maybe 30, 40 rounds. But crucial is to have very good seeds. Dream or.
Maybe also how long was the region? Also, it's shorter or longer.
Yeah, the length of the sequence.
Right, but we didn't.
Okay. All right, I think we'll have to take the rest of the discussion.